Thank you very much indeed, Max, for that kind introduction. And good evening, everybody. I'm delighted to see so many people here and to learn that you have this interest in global deworming. I'd also like to thank both societies, the Royal Society of Medicine and the Linnean Society, for this honour, really, of being able to give the Darwin lecture. When you think about Darwin and worms, one of the things that comes to mind, or you might be aware of, is this caricature, this cartoon that appeared in Punch many years ago, 1882, which was actually says at the bottom there, man is but a worm. And it was really a little ridicule of the idea of evolution, which we now have come to accept. And Darwin, of course, had a great love of worms. He did a lot of work on earthworms. So I thought it'd be good just to put up a strong, succinct statement as to where we are, in that nothing in biology makes sense except in the light of evolution. So I think that's a good starting point. And I also thought, as it was a Darwin lecture, I'd take a little bit of a liberty and put another picture of Darwin up, because the more somber figure on the left of the screen was actually drawn by my great-grandfather, so Harry Furness. So that's an opportunity for me. So what are we going to be talking about? What's the outline? What I want to do is just briefly talk a little bit about the Linnean Societies here and look at some of the early parasitological records. I will then want to introduce you to the worms and these things we call neglected tropical diseases. How are we going to control and eliminate the NTDs caused by worms? And then I want to take you on and introduce you to a very successful program, an eradication program called Guinea worm. And we were almost there with the eradication of Guinea worm. It's a tremendous success, but there are now some surprising new challenges which I want to touch on. But I also want to go then on to a different disease, my own favourite, schistosomiasis, and look how selection pressures, the kind of control interventions and the environmental changes, what's actually happening in terms of the schistosomes and snail populations that we study, and what's going to be the impact on our control and elimination efforts. So it's fascinating when you look back at the records of the Linnean Society to actually see this very strong association with the society and parasites. Linnaeus himself actually named Taenia solum, that's the pig tapeworm, and then some 30 years later went on to write a review of Taenia, so that's great. We have medical input there from Sir Anthony Carlyle, who made some really interesting observations on the tapeworm, in that he recognized that the tapeworm had this ability to be very prolific, to lay loads of eggs, which allowed it to become established and to continue its life cycle. So very early observations, and they're embedded here. And there is a display up in the library, which I hope we will have a look at, which is really, really quite fascinating. Many other connections there. Trichinella spiralis was in fact some connections to the museum, named, not discovered, but named by our first director, Richard Owen. And then if you look into 1864, work on the structure and nature of Guinea worm, which I'm going to talk about a little bit later. And all this is down to a previous executive secretary, it's well worth going back and having a look, who did some detective work about the society and parasitology. So that's the setting for the Linnean Society, but what about the worms? What are these neglected tropical diseases? What are we talking about? What's the impact on the global health burden? And we talk about neglected tropical diseases, but not many people really know what diseases are encompassed by that term. Thing that was brought together to actually cover 17 diseases which are very prevalent in the tropics and the subtropics, particularly in Africa and Asia and parts of South America, but they're diseases of poverty. They're diseases where people are living, where there's a lack of sanitation, and they have close connections with infectious vectors, mosquitoes and biting flies, and other infected animals. NTDs are not the big three. They're not don't include malaria, TB, HIV, or AIDS. And why are they neglected? Why have these diseases been neglected for so long? Mainly because they're hidden away in rural communities, resource-poor communities, as I've said. They're diseases with long names: onchocerciasis, leishmaniasis, trypanosomiasis. They're not diseases which are commonly recognised by the general public. Nevertheless, they're extremely debilitating, and they are diseases which can lead to a lot of social stigma and indeed can lead to huge mortalities. Indeed, it's estimated that NTDs actually kill over 500,000 people worldwide every year. So these are serious diseases in our global populations. And they're common. We have 149 countries with at least one of these diseases, and many places actually have more than one of the neglected tropical diseases, and most infected individuals will probably have more than one parasite actually causing the infection. Now, there's some good news in that the treatments are available for these diseases, and we do this by a mass drug administration program that's actually making sure that diseases are delivered to school-aged children, to communities where they're needed. We call it preventive chemotherapy, mass drug administration, and the costs can be quite low. So it's estimated that to treat a child or a person could be as low as 50 cents per year. So let's just have a look briefly at these worm infections. Some of the names I've mentioned: lymphatic filariasis, transmitted by mosquitoes. It's a nematode worm. It blocks the lymphatic systems, where you get swollen limbs, severely disfiguring and very debilitating. Schistosomiasis, I'm going to talk a lot about. The picture there on the top right-hand of the screen. There we've got massive liver enlargement due to the deposition of parasite eggs. And this isn't an old textbook picture that I've pulled out. This is a picture that was taken this year. So these kind of cases still actually are with us, and with us in a very big way. We have then a disease many people will know about, river blindness, onchocerciasis, transmitted by that tiny fly, the black fly. And then we have the guinea worm there on the right-hand side, which is transmitted, as we'll see, by copepods in fresh water. So all those diseases require a vector. They don't go from child to child or person to person. They need to go through a vector to actually continue their life cycles. The soil-transmitted helminths, other worm infections, Ascaris, Trichuris, and hookworm, are intestinal parasites, and they don't need a vector to continue their life cycle. They're either transmitted by the eggs, or in case of hookworm, by penetrating larvae. But look at the number of people infected. We've over 1 billion people actually infected with soil-transmitted helminths. And when you do treat children and the eggs are expelled, this is an extreme case, but this shows you how nasty some of these infections can be. And indeed, they do cause health problems, lack of attendance, cut down on school attendance, cause stunting in growth, cognitive development, all kinds of things associated with some of these infections. And clearly, you're a lot better off without them. So what are we actually going to do? What's actually happening to fight NTDs, these neglected tropical diseases? What's the situation? What can be done in these kind of places? And this shows the picture here is a treatment program going on at the school level. And then again, there's good news here that in 2012, here in London, there was a meeting which is now embedded in the history books, really, which was now called the London Declaration on Neglected Tropical Diseases. This was inspired by the WHO. They produced a roadmap to say that, well, we have got the tools to do something about these diseases. Let's actually try and do something. And they set a timeline, 2020, to try and eliminate 10 of the devastating diseases here. But a lot of the effort here was due to this man, Bill Gates, because Bill Gates was able to bring together 9 CEOs of pharmaceutical companies to actually sit together in the same room. They don't normally do this, don't normally cross the street to speak to another, but Bill managed to get everybody into the same room to say, look, let's all get together and work together to see what we can do. And that's what's happened, in the sense that we now have some very big and generous donations from the pharmaceutical world. And I think we should just pause to recognize that. It's not often mentioned that pharmaceutical companies are doing a good job in giving donations. There are a number of examples. I've just pulled out 3, which are relevant to what I'm talking about. But GSK, GlaxoSmithKline, have already donated about 2 billion tablets to treat lymphatic filariasis using albendazole. Merck Serono have currently upped their production of the drug for schistosomiasis to 250 million tablets a year. That's a lot of tablets, and that actually will treat 100 million children. And these drugs are being donated. They've donated to the countries that are requiring them. And there's another example, Johnson & Johnson, again for intestinal worms and albendazole, 200 million tablets per year to 2020. So there's a lot of drugs being donated, a lot of drugs coming into the system, but of course you've got to implement the control. You've got to actually get those drugs to the people that need them, and that in itself is quite a big challenge. So where are we going? How are we doing with this push towards elimination and delivery of drugs? This is a very recent slide from the World Health Organization. These are diseases at the bottom: lymphatic filariasis, onchocerciasis, soil-transmitted helminth, schistosomiasis, and trachoma, which we're not talking about today. But if we just take schistosomiasis, we can see, well, we're not doing too well, actually. It's a little bit, we're only treating, so far, treating about 30% of the people who actually need treatment. And that's a bit of a shame, but it's a case that we need more resources and more activity to actually get drugs. We now have drugs available, so we need to bridge this gap to get up to what we call 75% coverage so that we can get on the road in many different places to elimination. But there are much better stories with onchocerciasis, much better stories with lymphatic filariasis. And I think it's encouraging because over this time period, we can see that the donated drugs, the coverage that we're achieving in Africa and elsewhere is improving considerably. But of course, there's a downside. You deworm the child, or you deworm the members of the community, but lifestyles don't change, water and sanitation doesn't change overnight, so after deworming, we get rapid reworming. And unfortunately, that's why with these programs, we have to go back, at least on a yearly basis, to treat people in these situations. So that's the kind of where we are with the push into control. And it's what I've spoken about really is all about chemotherapy. It's about using drugs to treat these infections. But of course, these parasites have got complex life cycles, and so there are other ways that we can actually interrupt them. But we might just pause and think, well, if we're doing these additional interventions, what selection pressures are we actually imposing on the parasites themselves? The first one that might come to mind, well, parasites might become resistant to the drugs. If we're using and increasing our drug use, then we may see resistance. Certainly, as we all know, our vectors, our mosquitoes, our biting flies might become resistant to the insecticides that we're using. We may see some host shifts. We may see parasites jumping from one particular host, from man, or from a reservoir host into man. We've got good examples of that with the viral infections. We've seen it with Ebola and HIV and even malaria. We may see behavioral changes because of these selection pressures. We're beginning to see that mosquitoes, which bite at night when we have bed nets there is now increasing evidence that we're getting more day biting for mosquitoes because of the need of them to actually feed. So all these kind of pressures are going on at the same time as we've got environmental changes, we've got environmental precipitations, and we've got climate change. So it's a very complex kind of picture to deal with. So let's move to our first successful, really good example of guinea worm, where this could be the next disease to be eradicated after smallpox. Great steps forward being made here. What is guinea worm? It's a worm we call Dracunculus, and it's a nematode. It's a large nematode. This is a female worm being extracted from the foot of an infected individual. The worm comes to the surface of the skin. It can be very long, over 1/2 meter. It comes to the surface of the skin, forms a blister, burning sensation. There's a real need to put your foot into the water to cool it down. When that happens, the worm pops his head out, or her head out, and releases larvae into the water. So to capture this worm, you have to do this kind of technique, catch it on a stick and roll it round, and slowly pull it out and hope that it doesn't break. When the larvae actually come out of the worm into the water, they may be ingested by a copepod. And here, if we look carefully, we can see a copepod and a squiggly thing in the middle there. That's the larva inside. And if that copepod gets into drinking water, is ingested, the life cycle of the worm continues. So what can you do? Interestingly, I'm telling you this is the most successful control program. It's one that hasn't actually involved any chemotherapy. It's one that's actually benefited from the interruption of the life cycle primarily by filtering water and also treating water in some cases to kill copepods. But here we have a simple drinking device. I don't anybody would really want to drink out of that water, to be honest. But it's now going to be safe water in terms of guinea worm because the copepods are not going to be ingested. On the right there's a poster. I took that last year from Côte d'Ivoire. It's actively going on. It's to show people what the worm looks like and also to give a monetary award for case reporting. If you see a case like this, well, report it to the health centre and you get quite a nice bonus, 50,000 CFA, which is quite acceptable. So people begin to be aware and begin to look, see whether these infections are actually can be found. So this is, as I said, it's a good story. 1986, we had about 3.5 million cases of dracunculus, and now we're down to 22. From 21 endemic countries down to 4. This is really good news. Everything seems to be going well, and there was a lot of confidence building that there was soon going to be the announcement of the eradication of guinea worm. And I should add here that there are a number of foundations involved in this attempt for eradication. The Carter Foundation has been very active in this area, together with others. So there we're down to these very low number of human cases, and in Chad, 9 human cases, and then wow, suddenly discovered dogs are infected. 508 dogs infected in Chad. That's not elimination, that's not eradication, that parasite is actually being transmitted. The same kind of situation occurs with a dog. Here we can see a poor dog with his foot in the water and the worm being actually extracted. So what's actually happened here? Is it a host switch? Is it moving? Is it the parasite jumping out of people into dogs? Is it the same species? Is it the same genetic strain? So we and others at the Sanger Centre are interested in actually doing genomic work, typing these parasites to see whether in fact they're the same genetic strain in the dog as we guess in the human host. One idea is that in fact dogs are eating the intestines of fish that are caught. When the fish are caught, the insides are cut out, thrown away, and it's thought the dogs might be eating those intestines containing the copepods, and so this infection continues. That remains to be seen, but that is a possible route for infection. But breaking news, a paper just out, 2016, not just fish involved here, but it's shown that frogs are now playing a role in the transmission of this disease. Infected frogs have been frogs have been experimentally infected, and now they're being seen with the parasite in natural settings. So we do need to know a lot more about the biology of the disease and what's actually happening before we think that we've achieved eradication. Okay, that's guinea worm. I'm going to swap parasites. I'm going to jump to schistosomes now, and the schistosomes are much more interesting because they have these complex life cycles. Not only do they go in the mammalian host, but they go in a freshwater snail as well. And it's that going into the freshwater snail that makes life difficult. Here we have a picture of adult schistosomes. They're about a centimetre long, and they're living in the blood. We can see here, hopefully, some parasites. This is a cow at slaughter, the mesenteric veins of the gut. There's a worm, there's a worm. When the female, and this is the female in this gynecophoric canal of the male, they're unusual trematodes because you've got different sexes, but this female is very much an egg-producing machine, and she needs to get up to the surface of the gut wall to deposit eggs, so the eggs actually, in this particular case, come out with the feces. We've seen this kind of picture before. This is a rural setting where we can see schistosomiasis. This is urogenital schistosomiasis. We get this gross hematuria, these very bloody urines we can detect with diagnostic strips. And this is what the egg of the parasite actually looks like. As well as the human host, we have the snail host. So if we go back to that water spot we found looking at earlier, we can upturn the leaves and find these small brown freshwater snails. And they're the snail host, the intermediate snail host for the schistosomes. So here we have a different snail. This is a snail Bionflaria in culture, in a pot in the laboratory in water. And you can see some of those little white spots floating around. These are the infective stages of the parasite. This is a stage that would actually burrow through the skin. And this is from our facility at the Natural History Museum called SCANS, Schistosome Collection Natural History Museum. Now we've got a close-up of those cercariae. They're very interesting. You can see they've got a head. They've also got a tail. You can just see the tail there. It's forked. They're swimming tail first. They're going through rapid periods of motion. It's a non-feeding stage. It's emerged from the snail. It's got to find the mammalian host. It's got to get onto the skin. It's attracted by lipids. Once it gets onto the surface of the skin, it's going to penetrate in, and the life cycle is going to start again. Here we have an egg. It's a slightly different egg to what we saw. This is an egg of the intestinal parasite. It's got a lateral spine. This is another larval stage, a myricidium, about to hatch out from the egg. There he goes. Off he goes. You can just see it. We're just going to catch it again here. Rapidly moving around. This is, again, it's a non-feeding stage. It's living in water. And the myricidium is hatching from the egg. And that's got to find a snail. And if we look at this, it's got a very complex surface. It's got a ciliated surface. Rapid movement of cilia driving that larva through the water. And it's got an apical region at the top, which enables it to penetrate into the intermediate snail host. And there we end up with those adult parasites. But what a complex organism. It's living in man. It's living in that hostile environment in the blood as those adult worms. The freshwater stages, we have two stages. We have the myricidium, we have the cercariae, the myricidium going into the snail, the cercariae coming out of the snail. We have lots of different morphological stages within the snail. So a very complex parasite. What I want you to remember is that within the snail, we've been doing this kind of study in Zanzibar, which is islands are just off the, many of you might have been there. It's a good holiday destination, as long as you don't go in the rivers. Just off the coast of Tanzania. We've been working on both islands, in Guja and Pemba, trying to see whether if we can actually eliminate this particular form of the disease, the urogenital form of the disease. We have different snail hosts, but we have one which is important in transmission. And where the dark blue is, we don't get schistosomiasis because this snail doesn't live in those kind of habitats down in the south of Guja. So what can we do as well as schistosomiasis treatment? Here we've got the treatment of the child with the drug praziquantel. Just to mention, it's a safe oral treatment. It's got downsides in that it doesn't kill developing worms, it doesn't kill juvenile worms, and it's got downsides in that it's a large torpedo, chalky tablet that's no good for the under 5s, for the preschool age child. So Merck and others are working on a pediatric formulation, but we know from the work of Russell, who's in the audience, and others, that schistosomiasis in the preschool child is very important, but at the moment that's a sector of the community not actually receiving treatment in most places. Going back to Zanzibar with our surveys, what can we do? we survey our school-age population. The population in Zanzibar is about 1.3 million in total. We're working in the schools and we're working in different districts, looking at different interventions. This just gives a bit of a pictorial thing of the kind we're doing. Collecting urines, examining urines in the laboratory, looking for eggs on filter papers to see whether they're positive, and doing quite extensive questionnaires with the local community to see what's happening. We're using Snail Control to actually break that life cycle, because remember, if we can do something with the snail, we can actually begin to control the disease. This isn't a particularly good method of snail control, because you're using a chemical which does have an environmental impact, unfortunately. It does kill fish as well as kill parasites in the water. So we have to be very careful where we use this. But if we can use it focally in places where people are making water contact, we feel we might be able to make some progress. We're doing a lot of work with the communities, with the school-age children especially, both in the public schools, the state schools, but also in the religious schools and the madrasas. It's an Islamic country. And we're working to see whether we can introduce safe play opportunities for children so they don't go into the water, to actually get the message across about Kichocha. This is schistosomiasis. Here we've got a group of children actually pretending to be the cicary in the water as they go along. Here we've got a tug-of-war game, and if you lose the tug-of-war, you have to answer 5 questions as to why you fell in the water and you got schistosomiasis. Lots of things like this. So we know that knowledge is improving, but the trouble is, many times, even if you've got the knowledge, you might not have the facility. You might still not have the clean water and the safe water. So we have many examples from drawings, from interviewing and talking with children, that this is still a problem in many areas. So yes, we do our madrasa-based Kichocha education, classroom-based, outdoors. We tried to introduce simple urinals, because it's a urinary disease here. If we have urinals, will people use them before they go into the water and things of that sort? That was a failure, I think. We conclude. A big success story has been the community laundry platforms. We built platforms in the different districts where we're working in the randomized study for women to actually wash their clothes. They're next door to well water or to pipe water. They come there, wash their clothes, they keep their children with them. They're not going down to the river to wash the clothes, and we think that may well be having some impact. So these are different kind of interventions that we can introduce. I haven't got time tonight to go into detail about this, but we're slowly seeing a fall in the prevalence, the number of children infected in this case. Not high in the first place, but now in this particular example, for the whole of Zanzibar, using chemotherapy and snail control, infections are coming down. And importantly, these high infection intensity, that red part of the graph, is getting narrower. So some progress being made, but gosh, it's a complicated thing. It's not easy to do. We've done 10 rounds of MDA now, 10 rounds of treatment for the schools and for the communities, but we appreciate we still have quite a long way to go. We know that there are areas which we call hotspots, areas where it's very difficult to break transmission. And one of the problems that we're now seeing is, in fact, when you get down to very low levels of infection, you need better diagnostics. You can't just rely on screening urines. You need to look more closely as to what's happening. We've been working with partners from Leiden, Govert van Dam and others, with a new diagnostic test. It's very sensitive, it's very specific, but it's not particularly good news, because if we look at the blue bars here, these are lots of different districts of Pemba, where we've been working, Shears, where we've been working, the blue bars coming along. This is just looking at from egg counts. Then we just look at the red bars, where we're using this new diagnostic test, which is looking for antigens in urine. Wow, there's a lot more schistosomiasis there than we anticipated. So if we translate that to what's happening across Africa, we realize that we've still got a long way to go. Okay, so that was changes associated with a very neat system in Zanzibar, where we've got one parasite and one one host. So we now want to move from Zanzibar into the interior of Africa to Malawi, and just show you how the interactions and biodiversity really matter in terms of schistosomiasis in this particular place. Lake Malawi is interesting. It's a place many of you may have visited. Some people go there to look at the cichlid fish. But down on the south of the lake, here's some of the lovely, lovely fish that you can find. It's extraordinary, as many of you know, the diversity of cichlids you get in Malawi. There's believed to be well over 1,000 species of fish in the lake. Fortunately, there are not as many snails, but one of the interesting snails that's been found is this snail, Balinus naisanus, which is living in this looks quite pleasant place, but this snail's quite unusual because it's actually living on these sandy bottoms. It's quite different from that water lily environment I showed you earlier. But this snail is actually found living in these sandy bottoms, and is now playing a role in transmission of disease, which it may not have been doing previously. This is another snail, Balinus globosus. This is the typical habitat for globosus, not often seen in the lake, but more in the marshy water stream inlet areas where local people are getting infection. What's happening is interesting in that one of the cichlids, which is a mollusk-eating cichlid, it has this very interesting jaw apparatus which allows it to crunch on shells and eat snails, has been overfished. Basically, the cichlid in the shallow waters has been overfished by the community, some of them using mosquito nets, I'm afraid, but so the very small fish have been removed. And what this means is that the predator has been removed and the snail population has actually increased. And it's an unusual snail, but it is actually playing a role in transmission. So you've overfished, snail populations come up, transmission increases because of that. What might you want to do? You might want to actually increase that snail population. But here we can see the stomach contents of that particular cichlid, absolutely full of snail shells. So if you remove the predatory pressure, as it says here, you're going to have an impact on the snail population and on schistosomiasis transmission. So that biodiversity, that interaction between species in Lake Malawi is so important. And interestingly, Malawi is one place for tourists coming back to UK have often been to. That's probably the most common country for tourists to get schistosomiasis. Well, it was. I'll show you something else shortly. So if you remove the predator, predator, that can actually have an impact. So can you reverse that? Can you increase predation on snails? Can you actually begin to think about some effective biological control rather than using these chemicals? There's a very interesting example in Senegal and now in Niger of introducing this fearsome-looking freshwater prawn, which actually eats snails. It loves to eat snails because it changes its own shell carapace very frequently, and it needs a lot of calcium, so it's actively looking out for snails. And the good thing about this one too, it's also good to eat. So there are programs going on to see whether this can be introduced for biological control and also as a source of protein. Right, let's jump out of Malawi and move rapidly over to Senegal, over to West Africa, where now we're going to look at environmental change and also some interactions between parasites, which could be quite serious in the long term, because we're beginning to believe that we're seeing a zoonosis, infections in other definitive hosts, other mammals, actually evolving in this particular area. Many of us, some of us in the room have worked here. Joseph and Vaughan will know these places quite well. But the situation we're talking about is in Senegal over here in West Africa. And we're particularly interested in this region, the Senegal River Basin. Senegal River forms the boundary between Mauritania and Senegal. It was formerly a low area of transmission, not very much schistosomiasis there. But following droughts during the 1970s, it was decided, World Bank money and else thing, that a dam was to be built here, the Diama Dam. Another dam was built over here in Manantali. The one here in Manantali actually influenced the flow of water down the river. The Diama Dam stopped the salt water actually coming upstream. So it was actually to provide more fresh water for agriculture, stop that saltwater intrusion. This is the barrage. It's not a big dam, it's a barrage. It's just going across the river. That became operational way back in 1986. Previously, because it had been salty water, because there hadn't been many habitats for snails, snails were not abundant, but shortly after the dam was built, lots of irrigation canals, lots of agriculture going on, and snails moved in, and there was a massive outbreak of schistosomiasis. Massive outbreak of schistosomiasis. Some of the cases that you could see there, we've measured the number of eggs per gram of feces. We were seeing something like 9,000 eggs in just 1 gram of feces in some infected children, which the number of worms there would be unbelievable. These are some of the snails that thought it was quite a good place to live. We've got 5 different species of Bolinus forming taking different roles in transmission of disease. This is the one we saw in the video. This is the one that's responsible for intestinal schistosomiasis. That was the one that took off very rapidly and was responsible for the increase in intestinal schistosomiasis. A lot of studies are being done in this area. We did one looking at infections of urinary schistosomiasis, Haematobium, Schistosoma mansoni, in this region, Richard-Toll, and around this Lac de Guerre region, just to see what was happening with treatment. Could we, when we're using praziquantel to treat these heavy infections, what's actually happening? Because previous results have suggested that Schistosoma mansoni was not responding well to drug treatment. And this, unfortunately, is what we've found. This is a little bit of an outside now, but it's still the same situation, in that over 1 year period, we infected children who were infected with both Haematobium and mansoni. They got eggs in urine, eggs in stools. We gave them 2 treatments of praziquantel, spaced apart, so that we could actually knock out the adult worms and knock out developing worms. We came back after 6 months and did exactly the same 2 treatments, and this is the kind of result that we saw. With Schistosoma haematobium, that's a yellow bar, we can see we've got treatment, really good cure rates coming right down, and we would say there's good response to treatment, and then towards the end of the study, there's some reinfection going on. With this parasite in the same host with the same treatment, yes, we've got reasonably good cure rates going on, but rapid reinfection, so much so that after 6 months, we're almost back to where we started. Treatment again, not so good this time. At the end of the 12-month period, those children have the same level, there's the same prevalence of infection, and interestingly, the same intensity of infection. So it's beginning to suggest in this particular situation that we've got poor response to praziquantel with Schistosoma mansoni. And this is something that we need to monitor and follow biologically up. Interesting situation between 3 different parasites in Senegal. This is Haematobium. This is a urinary parasite, which we mentioned. That infects humans, and it infects this particular snail host, and the egg looks like that. But we have parasites we find in cattle and sheep. One we call Schistosoma bovis, which has got a much larger egg, and one called Schistosoma curasoni, which develops in a different snail host, but it's got an egg morphology like Schistosoma haematobium. And we need to use molecular tools to identify these different species. And it is a complex situation, because there we have our 4 different schistosomes. Here we have all the different species of snail that that we see. Some snails are serving as hosts for one parasite, other snails are serving as hosts for another. So, for example, Schistosoma curasoni are developing in Bolinus umbilicatus, but not particularly so in these snails up here. Haematobium developing well in globosus at that stage, and Schistosoma bovis developing in truncatus. Well, we began to start looking at these parasites in more detail, many of us in the laboratory, And what we could see was, in fact, some very interesting things going on between the parasites. What we're able to do is hatch the eggs, capture those miracidia in this kind of setting. We place them onto what looks like to be a filter paper. We call it an FTA card. But what this allows us to do is to actually look at the genetics, look at the gene sequences of the parasite when we get it back to a laboratory where we can, when we're able to do that. So we can collect eggs and miracidia from infected children, and we can also collect those cercaria from snails, and we can type them to see what's actually happening. When we do that, and this looks a little bit of a complicated tree, but what we're able to show, and Bonnie Webster, who's in the audience, was very much part of this with others, that we began to see that this parasite, Schistosomiasis, the parasite of man, was actually forming active hybrids with the parasite from cattle. So we had these hybrids which we could detect because of their both genomic sequences and also because of mitochondrial sequences. So here we have, for the first time, Haematobium and bovis forming hybrids in children. Now there's a question mark immediately. Will those parasites still respond to treatment? Will those parasites develop in cattle? Lots of questions going on. We're fortunate we've got another grant from BBSRC where we're following this more closely to actually see what's happening. This is a magnificent picture taken by one of the postdocs, Elsa Ledger, in the lab, which shows the situation and why we get the hybrids. Because here we have the cattle, and here we have the people, and also we've got donkeys and all sorts of things going on in the water. And this is where those parasites are actually mixing up, and we'll be seeing infections in these in people. The other interesting thing that's happening is that I said at the beginning that one parasite goes in one snail, the other parasite goes in another. The hybrid parasite actually enhances its infectivity and develops in both of those snail hosts. So we've got host switching going on in the definitive host, and if you look in the water, we've got host switching going on as far as the snails are concerned. As I said, we're following this up further. We've been doing a lot of surveys in different parts. Here we can see the worms again in the mesenteric veins. We can collect those worms. We can do our molecular diagnosis on these worms. We can collect urines. Here we can see some heavily infected urines. The hematuria, those urines are going to have the eggs. And this is just actually looking in the guts of cows, in the mesenteric vessels of cows. And we've got different study sites in Senegal. But what we're seeing is, in fact, we've got hybridization between Haematobium and bovis, and we've also got hybridization between a parasite, Schistosomiasis, that we see in a different area. So there's a lot going on there in Senegal. We've got agricultural development, we've got snails changing their distributions, we've got parasite mixing, we've got new parasites emerging which are infecting people, and we're testing to see whether they're also infecting cattle. We thought it was quite exciting at the time that you had a little bit of difficulty getting funding until, lo and behold, something extraordinary happened in Europe, in Corsica. We had an outbreak of urogenital schistosomiasis in Corsica, right down here on the Cavu River, a lovely place, been there myself. And this isn't your typical transmission shot because it's full of tourists. But those tourists, in fact, were exposed to Schistosoma haematobium. And what we found was, in fact, working with Jérôme Boisset and a whole host of people in Perpignan and also in Spain, that actually 120 tourists were infected with schistosomiasis. And that took a little bit of doing because the tourists only spent a little bit of time in the water. They went home. Some went to the doctors and complained of blood in urine or complained of abdominal pain. And of course, local GPs would say, well, that can't be schistosomiasis. You haven't been anywhere like that. But it was, and we were able to show that was the case. There were two main infection foci in the Kavu River. And what we were able to do was actually get some of the eggs and myristidia from some of the infected patients. And what surprised us was, in fact, that those parasites, when we typed them in the laboratory, they weren't straight Haematobium, they were hybrids. They were Haematobium bovis hybrids, and in fact, we had our first case of a straight Schistosoma bovis egg in one of the infected patients. Now, it's not particularly a surprise, I guess, because the snail, Balinus truncatus, has been there for a long time. It lives there successfully, and it has been for some time. But what's happened is, in fact, the parasite has been introduced into that environment, established, surprisingly, probably not a lot, but it shows the power of the parasite, if you like, that so many people became infected over a short period of time. And when we type the parasite, what happens is that we see that the schistosomes that we get from Corsica are almost identical to the ones we're seeing in Senegal. So what we suspect, and we're fairly sure of with further genomic work going on, that in fact the parasite that's ended up here is in fact the parasite that we've been seeing in West Africa. Quite sensibly, the French government took this extremely seriously and banned swimming in the area, banned the water contact, tried to forbid swimming there. Here we can see many of those snails collected in these kind of environments. That was in 2014. It's got a big impact on the tourist industry because people are going there specifically for these kind of water activities. When we look closely, this just shows some of the molecular kind of results of the phylogenetic tree. This is for the bovis component of the parasite. If you're any doubt, here we can see Senegal, and it's all embedded in the Corsica samples. So I think there's not a lot of doubt about what's going on there now. People are now allowed back in the water. Things are actually taking place again. Interestingly, in 2015, there were further 2 cases, but the situation has now been very closely monitored. Snails are being collected on a very regular basis to see if it could reappear. So, again, just from the Darwinian side, we've got to think, well, there are a lot of lessons here as to what's actually happening. There's also a question as whether schistosomiasis is really now a tropical disease or if it's in fact expanding its range. Because we do have climate change to worry about. We do have to think about snail distributions. We're finding these snails in Spain, in Sardinia, and elsewhere. We need to know a lot more about snail distributions. We have immigration. We have people moving both ways. A lot of French tourists going to Senegal and back to France, as well as from Senegal into France and from elsewhere. We've got to be aware of that, because when people move, they move with their parasites. And we've got, in case of Corsica, we've got the impact on the tourist industry. Okay, so I think that my time is up, but just a quick summary. I hope I've set the scene to show you that there's a lot going on with neglected tropical diseases. There's some very good stories in relation to Guinea worm. We are really close to global eradication. That's a massive thing. People might argue, well, be careful you save the parasite and try and do that in repositories. But other diseases such as schistosomiasis and all the ones I haven't mentioned in detail present a lot of great challenges. We've got to be aware of the genetic diversity of the parasites. They have complex life cycles. They will respond to new selection pressures, and we will see changes over time. Lots of things to take into account, loss of biodiversity, as we saw in Malawi, migration of people, invasion of new vectors, climate, environmental change. All of these things are going to have an impact on NTDs and on disease transmission. We've seen a little bit about new diagnostic tools. We've only got one drug for schistosomiasis, praziquantel. We really do need more. We need better tools. And the bottom line is that still, despite the huge donations, and the British government itself has put a lot of money into implementation of schistosomiasis control especially, there are insufficient resources for treatment and improvement of sanitation. So that group of people, that 1 billion number, which we call the bottom billion, are still in need. They still need resources to actually improve the situation. Lots of people to thank, particularly the people on the top in bold. David Molyneux, who gave me the updates on Guinea worm, Jay Stoffer and Henry Matzing, colleagues in Malawi, Amadou Garba, head of WHO NTD programs for the WHO figures, Jerome Brasset and his colleagues in Corsica, Joanne Webster and Nelson Ledger at RVC, all the people who did the work at the Natural History Museum, Aidan Emery, Bonnie Webster and Nick Gouvra, Fiona Allen, Steffi Knott and Muriel Rabone, and also to funders, SCORE, University of Georgia Research Foundation, Bill and Melinda Gates Foundation, the Wellcome Trust, have always been very supportive, and recently our friends in BBSRC. So thank you very much.